You know, we look at photos all the time, but what if a picture could be more than just a memory? What if it could be a piece of scientific equipment? Well, that's exactly what we're diving into today, this whole challenge of getting the green right. We're talking about the science of turning a totally normal photo into a super precise measurement tool.
All right, so take a look at this. It's a patch of grass. Looks pretty healthy, right? It's green, it's dense. Your eyes are telling you everything's great. But here's the thing. In science, looks healthy just doesn't cut it. That's not a real measurement. So the real question becomes, how do you actually quantify that greenness?
And that's really the heart of the problem we're tackling. How can we take a simple photograph, from a regular camera and turn it into precise, reliable data? How do we make sure those colors are spot-on accurate?
Okay, so could a camera be the answer for measuring grass health? To figure this out, some researchers set up a pretty cool experiment. They wanted to see if a totally normal, off-the-shelf camera could actually be trusted to measure how healthy the grass is.
So picture this. You're in a greenhouse, and it is just filled with hundreds of these turfgrass samples. Every single one needs to be evaluated. Now imagine doing that just by looking at them. It would take forever, and worse, it's totally subjective, right? What I think is a 7, you might think is an 8. The goal here was to find a way that's way faster and, most importantly, objective.
And this really gets to the heart of it. You've got the old way versus the new idea. The old way? Someone literally just looks at the grass and gives it a rating. Super slow, super subjective. The new idea? Hey, let's use a consumer camera. It's cheap, it's fast, but there's a huge catch. Can you actually trust the colors it's capturing?
So how do you get around that problem? How do you teach a camera to see true color? Well, it's kind of like you have to show it what true looks like in the first place before you can trust what it sees.
And the solution starts with this thing right here, an X-Rite color checker panel. Seriously, just think of it like a ruler, but instead of measuring inches, it measures color. Each one of those little squares has a very specific, scientifically known color value. It's our ground truth, our perfect reference point.
Now here's where it gets really smart. You take a photo of this color panel under your exact lighting conditions, then software goes in, automatically finds every single one of those squares and does a comparison. It looks at the color the camera actually saw and compares it to the color it should have seen. It's basically checking the camera's homework.
It really boils down to 3 simple steps. 1, you snap a photo of that standard color checker panel. 2, the software compares the photo's colors to the known perfect values. And 3, it creates what's called a correction profile, which is basically a set of instructions to fix all the color errors in any other photo you take.
And here's the proof in the pudding, right? Look at the difference. On the left, you've got the camera's original photo of the panel. On the right, the photo after correction. The difference is pretty clear, isn't it? It's brighter, the colors just pop in a more accurate way. The software has basically fixed the camera's vision.
All right, so the correction looks great on a perfect flat color chart. But what about on the real thing? How does this hold up when you're looking at the complex, messy, variable colors of actual grass? It's time for the correction showdown.
Okay, let's look. Left side, that's the raw, unprocessed image, straight from the camera. Right side, that's the same exact photo, but with our panel correction applied. It's definitely different, right? It looks a little brighter, the green is a bit different, but this is the big question. Is this actually the most accurate color?
But they didn't stop there. They decided to try boosting the saturation too, make the colors more intense. The thinking was, hey, maybe this will help the software do its job better. But you can see the problem, right? Does boosting the saturation actually help the analysis, or does it just change the data and mess everything up?
And of course, this is where things get messy. Because that's what science does. A new problem popped up. Even inside their special light box, the LED lights weren't perfectly consistent. As they warmed up, their brightness would change, just a tiny bit. But that tiny variation could be throwing off the very data they were working so hard to correct.
So now we're putting it all to the test. With all these corrections and potential problems, how do you even know if you're making things better? Well, you need a second opinion, a totally independent, trusted measurement to compare against.
And that second opinion has a name, NDVI. It stands for Normalized Difference Vegetation Index. But don't worry about that. All you need to know is that it's a super common scientific measurement from a specialized sensor that tells you how healthy a plant is. It's basically our gold standard.
So this scatter plot basically compares the camera data to the NDVI data. Simple enough. Each little dot is a grass sample.
What we're looking for is a tight, straight line. The tighter that line, the better the correlation. It means the camera and the special sensor are telling us the same story.
And check this out. At first, it looked like a massive win. For one of the main ways they measure greenness, an index called DGCI, the correlation with NDVI shot up by over 21% after the color correction. I mean, a 21.4% improvement. That sounds amazing, right?
But yeah, it wasn't that simple. While that one metric got way better, a couple of other important metrics actually got worse, like a lot worse. One dropped by 11.5% and another by almost 9%. So the correction was helping one measurement while actively hurting others. What's going on here?
So we're left with this surprising, really complicated verdict. The experiment didn't just give a simple thumbs up or thumbs down. The answer was, well, it was messy.
And this is why it matters. The whole point is to rank the grass from healthiest to least healthy. Well, because of the correction, this one sample right here jumped up 3 spots in the rankings. Think about that. These little digital changes were actually changing the final results of the experiment.
And if we step back and look at the big picture, you can really see this weird push and pull. The correction made things way brighter, up 26%, but it also crushed the saturation by almost the same amount. The overall correlation with NDVI, it did get better, but only by about 3.5%, so not exactly a home run.
And the researchers themselves said it best. They basically said the results were inconclusive, which leaves you with this classic science problem. Was NDVI, their gold standard, actually the wrong thing to compare against, or were the color corrections just not really working consistently?
So when a scientific experiment gives us a messy answer like this, does that mean it failed? Not at all. I think it means we've just discovered a deeper, more complicated truth. What seemed like a simple idea, let's just fix the color in a photo, turned out to be way more complex. And that's what's so cool about science, right? Sometimes the messy answers are the most important ones, because they show you that the real story is so much more interesting.